hydrogen bonding. Hydrogen bonding is extremely important in biology because everything in biology is filled with water or surrounded by water. And hydrogen bonding is very common between water molecules and can be a very strong force. So we have to get familiar with how that's going to work. So hydrogen bonds happen because it's a bond between oxygen and hydrogen. Hydrogen has a very low electronegativity, and oxygen has a very high electronegativity. So we're going to get a very polar covalent bond here. And with hydrogen only having one proton, so if hydrogen has only one proton, what's its atomic number going to be?
1.
Yeah, great. So with hydrogen only having one proton and one electron, when this polar covalent bond forms, you're going to get that electron to be mainly near the oxygen and not near the hydrogen, leaving just one bare proton floating around, or on the side of the water at least. So we have kind of a bare proton here, which is nearly a full positive charge, and then we have a negative charge over on the oxygen, and that difference in charge is going to cause an attraction, and that attraction is the hydrogen bond. So the hydrogen bonds are much weaker than the ionic or covalent bonds, but there can be a large number of them, and they can form at energy levels that are really relevant to biology. So when we talk about how different molecules are going to arrange themselves and fold and unfold, a lot of that's going to be dictated by hydrogen bonds. Any questions on how this works? Again, it comes down to the difference in electronegativity between oxygen and hydrogen forming a very polar covalent bond. And there's going to be a lot of these polar covalent bonds in water, and they're going to dictate water's properties. All right, so we've been talking about how to predict polarity. So let's look at, this is olive oil here on top. This is the chemical structure of olive oil, and this is water. So is olive oil going to be polar or nonpolar based on these electronegativities?
It's going to be nonpolar.
Nonpolar, great, because it's mainly carbon-hydrogen bonds, and carbon and hydrogen have similar electronegativities. So olive oil is nonpolar. Water, on the other hand, we know is polar, great, because oxygen and hydrogen have a large difference in electronegativity. So that means we have a nonpolar substance in olive oil and a polar substance in water, and when we put them together, the olive oil? Yeah, so we know it's, olive oil is nonpolar because we can tell from the structure that it's mainly carbon and hydrogen bonds. And carbon and hydrogen have similar electronegativities, so they're both going to pull equally on the electron, and they're going to share it equally. It's like if there's a tug of war contest. If someone's way stronger, they're going to get more of the rope. So we have a nonpolar substance that's not mixing with a polar substance. Are there any ideas why that might be?
Olive oil is thicker than water. I don't know why, what makes it thicker. It's the mass.
Right. So that would explain why it's like kind of towards the top, but it wouldn't explain like this really clear cut here.
Because the hydrogen.
Sorry.
The hydrogen
Not
bond.
pulling against.
Wait. Let me.
The lack of hydrogen bonding.
Yeah. And what do you want to say?
Because the hydrogens are not pulling against other hydrogens.
Right.
When
So
you mix oil and water.
yeah. So there's not much hydrogen bonding in olive oil, and there's a lot of hydrogen bonding in water. And with the hydrogen bonding comes water molecules want to stay near each other because they're both pulling on each other. And they can't really form any hydrogen bonds with olive oil because it's so nonpolar. So that means we have a layer of water here that's kind of excluded all this nonpolar substance.
Is that because electricity is higher in oxygen and it pulls stronger
Right, exactly.
than the carbon does?
Well, yeah, exactly. So the oxygen is going to pull more of the electron from the hydrogen here than carbon is from the hydrogen up here. So that's why, like you said, this is going to be nonpolar because they're pulling equally, the carbon and the hydrogen. Great. So, but the important thing is that we see that if you mix polar and nonpolar substances, you can get this exclusion, and that's going to be important also when we look at how things we dissolve in water are going to behave and form around each other. All right, so this would have been much better if this had ended where lecture was on day one. We're not going to go over this now because we just did a review, but I didn't want to cut it out of the slides because I think it's useful when you're reviewing for quizzes and exams. These are the types of questions that you might see. So on your own, you can go over these and look through the answers. And if you have any questions, let me know, but we're not going to go over them in class today because we just did a review. So we're going to start the learning objectives. So we're going to start the second half of Chapter 2. These are the learning objectives for today's lecture in Chapter 2. So we're going to want to look at water's unique properties, which mainly arise from hydrogen bonding and this polar covalent bond between oxygen and hydrogen, and what those unique properties do to its behavior. We're going to talk about acids and bases, and these are properties of solutions in water. That's going to be important for understanding how different molecules are going to behave in water as it pertains to biology. And we're going to talk about buffers. So buffers are what's going to kind of counteract the effects of acids and bases on a lot of your biology and keeps you happy and healthy. All right. So some of the chemistry of water. So like I said, water is really abundant in biology, and it's crucial to the way all biological systems work. So we have to really think about its behavior in order to understand bigger systems. So all of these properties of water that are unique compared to a lot of other solvents or liquids that we can see on the planet, all of these unique properties arise from its difference in electronegativity between the oxygen and the hydrogen. So its molecular structure leads to all these interesting properties. So we're going to focus mainly on the fact that water is a universal solvent. So if you had the lab already or looked through the lab manual, you'll know that solvent is a liquid that we're dissolving something inside. So in biology, we have a lot of things dissolved in water, either in cells or outside of cells. And the fact that water can dissolve a lot of things is really important for biology. So why do you think it might be important that water can dissolve things in it for biological organisms? Why would that be a necessary property?
Oh, because you add acid to water and not wanted acid?
No, I'm kind of talking about how in biological systems, the fact that water can dissolve things, like why would that be helpful? Yeah. Yeah, right. So with the fact that a lot of things can be dissolved in water, it lets biology move things around easier. So when things are in a liquid form, it's easier to pump them through a large organism or move them around different parts of a cell. So without water being able to dissolve things, a lot of important biomolecules would be just in a solid form, and they'd be harder to move around. So we're going to talk about cohesion and adhesion, which I think you'll really enjoy. And there's a little illustration of how that leads to some cool behavior from trees. All right, so water is what we call the universal solvent, because water is really good at dissolving other substances. So the reason this happens is because water has these large polar regions, these polar covalent bonds, and as they dissolve substances like sodium chloride, which is just a common salt, it breaks up these negative and positive particles, breaks up this ionic bond, because here around, like say the sodium, which is going to have a positive charge when you split it from the chlorine, it can align the negative regions of the oxygen around the sodium to kind of replace that ionic bond. And so it kind of satisfies sodium more because there's more charge around it, more positive or negative charge around this big positive charge. And that's going to be called a hydration shell. So the water that forms around an ion like this is going to form kind of a shell like we see here. And when that happens, we're going to say we have a solution, because this is going to be really evenly mixed. All these sodium and chlorine ions are going to be evenly spread throughout the water. It's like a homogeneous solution, we might say, like a homogeneous solution. And so that solution makes it really easy for biological systems to move things around. And so that's going to be really important to us in future chapters. Any questions about that? It's kind of a lot of information. So the key point, water is really good at dissolving things, makes it a universal solvent, and that's a function of these hydrogen bonds that we're going to talk about later. Oh. So again, the thing about water structure is it's the difference in electronegativity between the oxygen and the hydrogen. Sorry, it's like really hard to use two laptops. I need to get an adapter. So when we talk about water dissolving things, we're going to use terms like hydrophilicity and hydrophobicity. So if you look at these words, maybe let's start with the word hydrophilic in the beginning. What do you think that might mean based on the structure of the word? Water. So hydro, yeah, that means water. What's that? Yeah, so philic means,
It likes
yeah, like philo means like to love something or like something. Yeah, what's phobia mean? Afraid. So we have something that loves water and something that's afraid of water. So hydrophilic means it loves water. Hydrophobic means it doesn't like water. So based on what we've talked about so far, what do you think would make something hydrophilic versus hydrophobic?
It doesn't combine.
Polar, not the polarity of the molecule, is going to dictate whether something's hydrophobic or hydrophilic. Really, hydrophilic is a synonym for polar. So anything that's polar is going to be hydrophilic. It's going to love water. It's going to like to be around water. Just like in the previous example we talked about sodium chloride, when those ions break up, they love to be around water because they're charged and they have a really high charge, and they like to be surrounded by these polar bonds, these hydrogen bonds. Hydrophobic, this is water fearing. This is like the olive oil. So when we put hydrophobic things in water, they don't like to be around the water. They don't like to be surrounded by these hydrogen bonds. Yeah, exactly. Hydrophobic is like oil. They like to be kind of excluded and on their own, so they're not interacting with the polarity of the water. So this is a way that we could explain this separation between the olive oil and the water. We have a hydrophobic region that gets excluded from the water and a hydrophilic region that stays around the water. Do you have a? Okay. Okay. So we just talked about water as a universal solvent. Now we're going to talk about cohesion and adhesion. So cohesion, sort of like the word cohesive, like to stick together. Water has a really high, it's very cohesive, so it likes to stay near each other. Like these hydrogen bonds that we've been talking about, they keep water near itself. But there's also an opposite property called adhesion, which is like adhesive, like to stick to other things. Water can also try to stick to other things rather than just to itself. So if you had the lab before, or you're gonna have the lab tonight, you saw water in a graduated cylinder, and you saw this U-shaped thing called a meniscus, and that's because the water is going to be climbing up the walls of the container because it likes to stick to it, and that's going to be adhesion. So cohesion is water sticking to each other, and adhesion is water climbing up the walls of a container. Oh, sorry. Good? Okay. So if we think about trees get all of the water from their root system, but they have to get that water up to the top, the highest branches, and they have no heart in them to pump the water up from the roots up to the top. So they're going to rely on this cohesion and adhesion to get water from the soil up through the root system and up to the top of the tree. So water is going to climb up the walls of the container, which is going to be the tree in this case. Water is going to climb up the walls of the tree, and as that water climbs up, it's going to pull other water up with it. So we have the adhesion on the walls of the tree, like inside of the root system and inside of the trunk. That's going to pull water up these channels in the tree, like you can see here. So we have the water is going to travel up these channels here and up the root system and up the trunk of the tree. And as it does that, it's going to pull other water molecules up with it. It's So that's going to be an example of cohesion and adhesion. All right. Kind of spoiled this one, but maybe it'll still be a good question. What's that?
You already told us?
Yeah, well, let's see if people are listening. So does water get pulled up by cohesion or adhesion? Adhesion. So water gets pulled up the bark of the tree by adhesion, but it pulls other water molecules up by cohesion. Great. So now we understand these three interesting properties of water. Water is a universal solvent, cohesion, and adhesion, and how they sort of are starting to relate to biology. All right, so now we're going to move on to talking about acids and bases. So these are types of solutions that water is going to be forming. So just like when we talk about water as hot or cold, there's this kind of spectrum of how hot or how cold it is. There's also a spectrum of like how acidic or how basic it is. So we know that water is made up of hydrogen and oxygen. Usually it's two hydrogen atoms and one oxygen. But if you have a lot of water, sometimes it's going to break up into just a hydrogen, which is going to be positively charged, or an oxygen and a hydrogen, which is going to be negatively charged. And how much it breaks up into these components and how much of them there are is going to depend on what you're putting inside of the water. And what you're putting inside of the water is going to dictate how acidic or basic it is. So it's a scale, just like hot or cold, just like we have temperature. We also have this scale of how many hydrogens or how many oxygen and hydrogen groups there are here, which we'll talk about in a bit. You're going to have a kind of a spectrum of concentrations of both of those. And so we're gonna describe that spectrum with what we call the pH scale. So the pH scale is going to be a description of how many hydrogen ions there are in water. So usually water is like basically neutral, and so we say the pH is 7, and that's because there's as many hydrogens as there are those oxygen and hydrogen ions. They're about equal, and we say that's neutral. But if the pH is really low, that would describe a very acidic solution, or one where there's way more hydrogen ions. So there's a lot of hydrogen ions, we have a really low pH. If there's not many hydrogen ions, we have a very high pH, and that's going to be a basic solution. So the pH scale is kind of interesting because it's what we call a logarithmic scale. So on the top we see this formula that has this word log. So I'm not sure if everyone's covered this in all their math classes yet, but what's important about a logarithmic scale is that 1 unit in pH is a 10 times increase or decrease in the concentration of hydrogen ions. So if we go from pH of 6 to 7, we're going to decrease the number of hydrogen ions by an order of 10. If we go from 9 to 8, we have 10 times as many hydrogen ions. So a pH change of 1 is a really big shift in the number of hydrogen ions. So let's see if we caught that. So what has more hydrogen ions, pH of 2 or pH of 4? 2. And how many times more hydrogen ions does it have than a pH of 4? Okay. Heard a lot of answers. I'm going to assume everyone got it right. So when we go from 4 to 3, we're going to go up 10 times. And then 3 to 2 is another 10 times. So from 4 to 2 is going to be 100 times. So, okay. So each one number we change in pH is going to be a 10 times increase or decrease. Yeah. So as we go up 2, it's 10 times 10, so 100. That's what logarithmic means. And so it's a really good way to express a very wide range of numbers. Yeah. Do you have questions about it now, though? What's that? Okay. Let's practice a little bit more. So is it not clear what logarithmic means or what pH means? Because if we'll talk more about what pH really means in the next slide, but it's kind of important to understand the way we measure it too.
It's like a balance.
Yeah, okay. Yeah. So the thing is in solutions biology, what we see is like a really wide range of concentrations of hydrogen ions. And so we use this pH scale to explain that really wide range. And so what it means is each one number in the pH scale translates to a 10 times difference in the hydrogen ion scale. So it'd be like if we had another unit for currency and it was like say 2 coins was like $10 and 1 coin was only $1. And so it'd be like a scale. So if you lost a coin, you would lose like 10 times more than that. That's not a really good example. It sounded a lot better in my head when I started saying
Is it just like exponents?
We tried
Like
again.
every time?
That's great. Yeah, that was a much better.
10 to the second power, 10 to the third.
Exactly.
I got
That's exactly what it means. It's much. You should be teaching the class down there. Do you have a question? You won't have to use this equation at all. I just thought it would help guide your learning. So an example again though, if we go from pH of 7 to a pH of 8, we're gonna have fewer hydrogen ions, but how many fewer are we gonna have?
10 to the 1.
Yeah, 10 times fewer. Does that help? Yeah. Okay. Are there any more questions on that, or? I think if we move forward, it'll start to make sense.
If you go from 8 to 10, it's 10 to the 2.
Yeah, which is
100.
Yeah, great, good job. So we're interested in things that change the pH. So this pH scale is obviously really important to biologists because we're going to be using it a lot. And the reason it's important is because we see a lot of things dissolved in water, acids or bases, that are going to change this hydrogen ion concentration a lot. So if you put something like hydrochloric acid, this HCl, in water, what's going to happen is the H and the Cl are going to separate. So we're going to get a lone chloride atom over here, or ion over here, and the hydrogen is going to hop onto a water molecule. This is where it gets a little bit confusing. So you can either write hydrogen ion, just H plus, or H3O plus, because like the hydrogen is joined to water. So you see both kind of, you'll see both in your textbook, I think, both kind of ways of writing it, but they're really the same thing. So when we talk about hydrogen ions, it can also refer to H3O plus, which is just a hydrogen ion on a water. So when you dissolve hydrochloric acid in water, you get this separation of the ions, and you're going to get a much lower pH, because you're going to have a lot more of these hydrogen ions floating around. Whereas when you dissolve a base in water, like this, oh man, so, what is it again? I forgot, I remember this. Ammonia, I think. I forget, I'm sorry, I forget. But what's going to happen is this molecule is going to steal the hydrogen from a water molecule, and it's going to leave a hydroxide ion, this OH, so it's a negatively charged ion.
Are you talking about amino acids?
What's that?
Was it ammonia or amino acids?
I think it's ammonia, I should,
Oh,
amino, what? What is it?
I think it's
Ammonium. I was close. That was like kind of it. But what's important is that the bases are going to steal this hydrogen atom from water and leave a hydroxide ion. And this is going to have a much higher pH because there's going to be a really low concentration of the hydrogen ions. So as we put different things in water, we're going to change the pH of the water. And that's going to have implications for the way things are going to fold and unfold in water.
So the hydroxide ion is basically plus NH4. Is that what you're saying?
No, it's this hydroxide ion is going to contribute to the pH.
Oh, so
Yeah.
So that's how it'll look. Is that what you're saying?
What do you mean? So?
No. So you said like the hydronium ion, that's the H3 plus.
Yeah. Yeah.
So hydroxy, whatever, ion is that O.
OH minus, yeah. This.
Oh, okay.
Yeah, yeah.
So that means that's polar? No, that's not polar.
Yeah, no, these would both be polar because there's really big differences in the charge throughout the, yeah. Okay, so there can be these really wide swings in pH, and just like if there's really wide swings in temperature, you wouldn't really be that healthy. You know, if our bodies were kind of oscillating between like 20 degrees and 150 degrees, it would be really hard for us to live. So we need to live in places that kind of constrain the temperature shift, so we can be, you know, happy and healthy. It's similar with pH. Our bodies need a really narrow range of pHs to function, and if we're putting these acids and bases into our bodies just as a matter of living, we're going to have other systems that keep the pH in a really narrow range, so everything can function the way it needs to. And those things are going to be called buffers. So buffers resist major changes in the pH. So a buffer keeps the pH of your blood like near 7.4 all the time, which is like close to neutral. And if you didn't have buffers in your blood, your pH would range through a much larger, you know, kind of variation, like maybe between 6 and 8. And that would be really bad for a lot of the other molecules that are important for you to live. They wouldn't really function properly, and you wouldn't be able to survive. So buffers keep the pH of your blood and of your body in a really narrow range. So there's one buffer that's very common that we're just going to discuss briefly. It's called a bicarbonate buffer, and so your body is going to be able to control this buffer partially by adjusting the way you breathe and by your kidney function, which is going to release carbonate. And so we don't have to know all the details, but I just thought it'd be kind of useful for your background knowledge. What happens when we have a bicarbonate buffer here is that as we add or remove hydrogen ions due to putting acids or bases in the water, this buffer is going to be able to change that into either bicarbonate over here on the right or just carbon dioxide and water on the left. And so the buffer is kind of like a sponge that absorbs these changes in hydrogen ions and pH and kind of neutralizes them to keep your blood at an even pH. And so it does that partially through some chemistry that's really interesting that we won't really get into, but also by changing the way your body is actually going to function, by changing how much you're breathing or by changing what your kidneys are releasing. And so your body is really good at keeping your pH in a narrow range, because if it wasn't, you wouldn't be able to survive. So it kind of invests a lot of energy into the kind of this regulation of the pH. All right, I just wanted to include this link. We're not going to watch it now, but if you want some more background on buffers, you can look at that.
So when people say their pH balance is off, does that have anything to do with the buffer?
Yeah, so
what is the buffer doing at the moment?
well, if someone's pH is like not balanced properly, I don't really know too much about that condition.
That's something like sugar, red cells and white cells.
Yeah, well, I think it's like I could imagine it's just kind of like if your body is just not functionally properly anyway for some sort of like under stress, or you're not eating properly, or or something's not, you're not in optimal conditions. It might not be able to produce this buffer well enough to keep the pH in range.
Like you get respiratory alkalosis or acidosis.
Right. Yeah.
That's what the conditions are.
So there's like some underlying disease, and it's kind of throwing off the function of the buffer.
Yeah, and your kidneys have valve breathing.
Right.
Your lungs are compromised.
Right.
Well,
I forgot the chemical. There's your body releases. I don't know.
Yeah.
Tested by doctors.
What's that?
well,
In detail.
right, for the pH.
No. So what's going to be on the test is just the definition of a buffer.
Yeah.
Yeah.
Yeah, we'll have to.
Yeah. Okay. This is a review of what we've just gone through so far. So just think on your own for about 2 minutes, and then we're going to go over this as a class. So just try to go over. You can write it down in your notes if you want, but you should be able to kind of summarize these points. And if you can't, we'll go over it as a class.